the topic that we are going to start today. All of you are there? Fine? Free with brain? Free from brain? Brain is energized? Glucose is there in brain or not? No? Why you didn't have your breakfast? Late? Oh. You must have. Morning breakfast is very important for you guys, okay? So glucose is there in the brain or not? I'm just asking that. Does your brain neuron cells have glucose? Yes or no? नहीं है, अभी तो कोमा में होता है अगर नहीं होता तो अभी तो कोमा में चल गए होते. You would have been in coma. Okay? It is there, no doubt. What is a transporter? What is a transporter that is present in majority of the neuron cells? Transporter in neuron cells. I'm just revising the last class I have taken, glucose transporter. That is also responsible for adrenergic responses regarding specifically in hypoglycemic conditions. Anyone? GLUT1, GLUT2, GLUT3, GLUT4. GLUT4 is insulin dependent. It is present in skeletal muscle and cardiac tissues and adipose tissues. GLUT2. Okay, GLUT1 is predominantly present in RBCs. GLUT2 is predominantly present in liver cells and also to some extent in neurons. So what is the specificity? Km for GLUT2, affinity of GLUT2 is high or low? High. That means your neurons, your brain cells never gets deprived of glucose because the transporter that transports the glucose from the exterior, from the blood supply, from the blood capillaries from it to the inside of the brain cells, they have GLUT2 transporter. That means they have got higher affinity. Higher affinity bole to, even if the concentration of glucose is this much, very small, the glucose will definitely enter inside the brain cell. Okay? So that's why we never get deprived of glucose, even if the condition is hypoglycemia. Okay? So even if you have not had your breakfast, your brain is still getting glucose. Clear? So from today onwards, we are going to start with metabolism. So welcome to the most complex topics of biochemistry, that what you people are going to do, you are going to mug off. And whenever examination is there, we'll go out into the examination and vomit out. Only possible way. Fine? Am I true? No. These things should not happen. Because for understanding, for going through the metabolism, you need to understand few things. It's very simple. Believe me. But you have to follow the way of approaching metabolism. That I am going to teach you. You all are aware of glycolysis. You know what is glycolysis. You know that glucose converts to 3 carbon molecule of pyruvate. You know that. You know what are the steps. You have gone through that. Have you gone through? Yes or no? You all know. Then why to teach you that glucose ke baad ye banta hai. After that it gets formed. It is a substrate for this reaction, enzyme. This is the product of this reaction. You all know this. But you might not be knowing that how to approach a metabolism. So today I am going to teach you about that how to approach a metabolism. How to study. Not mugging it off. Rather than mugging it off, you can understand it. You can remember it by yourself only. Okay? So we will be having a different approach of teaching learning method today. Okay? Before, as I start, whenever you approach any metabolism, you must go through these questions. What, where, how, when? What are the questions? Whichever metabolism. You ask yourself, you specifically ask yourself, You specifically ask yourself these questions about that metabolism. What, where, how and when. Sorry. Okay. What is glycolysis? Anyone? Do you need to mug up first of all? Okay, fine. I have told you already that whenever you find any difficulty regarding understanding or remembering about the definition of any of the complex reactions or any of the complex metabolic pathways and all, I have told you what to do. Just split the word. Glycolysis means glyco stands for glucose. Lysis stands for splitting. Okay, fine. And whenever we talk about splitting, the process of splitting is always, that Uparwala is na, who has made us, is very uniform. He has never done partiality. We people do partiality. He has never done a single partiality with any one of us. Not even with our cells. So whenever we talk about splitting in chemical reactions, it has to be unpartial, non-biased. Fine. If a molecule is having 6 carbon, the splitting should be into 2 molecules of 3 carbon component. It has to be equal in both the ends. It cannot be partial. Okay? So lysis means splitting of molecule. Fine. Can you define it? Can you define anyone? I don't want bookish language or bookish statements. Anyone define glycolysis? Anyone? Just raise your hand and define. As you know, I am not going to scold you. What is glycolysis? Anyone? Just raise your hand and say no. See, I want my class to be interactive. मजा आना चाहिए क्लास में. ऐसा नहीं didactic lecture नहीं होना चाहिए. Yes, please. Breakdown of glucose into 2 molecules of 3 carbon pyruvate in the presence of certain enzymes. Fine, he is right. Okay, almost he is right. Is the definition complete? Incomplete. Fine, very good. You have added the location. Very nice. Okay, go on, sit down. Anything else? Still it is incomplete. Releasing energy. Very good. Some more points are getting added. Complete it. Glycolysis is a metabolic process that converts a 6 carbon molecule glucose into 2 molecules of 3 carbon pyruvate in the presence of enzymes that releases energy and that takes place in cytoplasm. Okay? Still it is incomplete. What is left, you say? The first statement, glycolysis is a metabolic process. There are two types of metabolic process, either anabolic or, so glycolysis is a process of catabolism. Is that clear? So see, you can prepare your definition by yourself. There is no need of going through the books. Okay, can we recite it? Can we recall it? It is a catabolic process that splits a 6 carbon molecule of glucose into 2 molecules of 3 carbon pyruvate in the presence of enzymes that are located in cytoplasm, and at last what you get? It causes the release of energy. Energy in the sense of ATP. Done. Okay. If you go through, glycolysis are of 2 types, aerobic and anaerobic. Okay. Whenever we talk about aerobic, presence of oxygen, and anaerobic, absence of oxygen. Yes or no? Fine. In presence of the condition called as aerobic, we get a product called as pyruvate. In absence of oxygen, we get a product called as, very good. Can anyone say why this difference?
The lactic acid is formed.
I know. Lactate is what? Lactic acid. Lactic acid का conjugated form का नाम ही lactic acid है, lactate है. Fermentation. Why fermentation is needed? Question aaya na? You got a question in your brain? Have you gone through this? Anyone who knows why lactate or lactic acid is formed in the presence of anaerobic condition provided by glycolysis, provided through glycolysis? It is very common in RBC. Remember, question aaya na? Okay, fine. We all will be remembering this question. याद रखिएगा. We all will be remembering this question. We will go through the answer of this question at the end of this topic. Okay? If I forget, please make me, remember this. Okay? Chalo. Where does glycolysis takes place? Whenever you answer this, the answer should be specific. It should be at the cellular level, at the cell level, at the tissue level. Okay? If it is at the cellular level, glycolysis takes place in cytoplasm. Does it take place in mitochondria? No. It takes place only in cytoplasm. Why? Why? अरे बेटा बाद में लिखना. बाद में लिखना. These all things are available in book. You listen to me. Why it is available in cytoplasm? What we call there? Is there any specific terminology? Yes, because the enzymes that are responsible for catalyzing the process of glycolysis is present in cytoplasm, and that terminology, what we call it, is compartmentalization of enzymes. What we call? This term is going to come for a lot of time. Okay? So the reason why glycolysis takes place in cytoplasm is due to the presence of enzymes specific for glycolysis is there in cytoplasm, due to the reason of compartmentalization of enzymes. This compartmentalization process or this compartmentalization topic is going to be clarified when you will go through protein trafficking. Okay? That protein is formed in rough endoplasmic reticulum that goes to Golgi bodies for packaging and all sort of thing, modification and all sort of thing. Fine. Then it goes to, they are packed into certain vesicles, they are sent to the different or localized part. This sending of a matured protein from one area to different area dependent on their work, dependent on the performance they are going to, the work they are going to perform is called as protein trafficking. Okay, this is a very major topic. Lot of diseases, lot of mitochondrial diseases are due to, lot of genetic disorders are due to disorders in protein trafficking. Fine. अब तुम्हारे घर पे जो पार्सल आना था वो मेरे घर पे आ गया. What will happen? Massive disorder. She ordered some lipstick that came to my room. मैं क्या करूँगा लिपस्टिक का? Massive blunder. So this happens because of protein trafficking. Okay, disorders of protein trafficking. So whenever you are going to write a note, if they ask you, then it should be of at cellular level, at tissue level. Okay, for all metabolic pathways. How stands for that? What are the steps? Okay? When stands for regulation, that we are going to conclude in the next class. May not be possible in today's class. Okay? Any doubt till here? Any doubt? Remember this. I want you people to ask me this. Sir, why anaerobic glycolysis may lactate formed? I want you people to ask me. Okay? Remember this. Is that clear? Chalo. Glycolysis starts from, whenever I'll be using red pen, remember that step is important. Okay? Otherwise I won't. Glycolysis starts from, glucose comes into the cell provided by transporters, GLUT2, GLUT4, whatever. Yes or no? Okay? Glucose is converted to glucose 6-phosphate. I am going to write as shortcuts. You should not be. If generally, one more thing, as I have told you that in the last class. Nowadays, according to the modern CBME and the preparation of questions and all, earlier days, earlier days, cycles were very important. You need to recite the cycle. They used to come in as a direct question in the examination. Direct question. Describe the complete process of TCA cycle. Describe the complete process of glycolysis. Nowadays, it has gone. They won't ask you the direct process. The question is never going to come from direct question basis. Rather, they may ask you some important part of this. They will more focus on some disorders related to this. They may focus, the question may come as a focused part regarding the regulation of this. But they won't ask you the direct question as describe the entire steps of glycolysis. No, it is not going to come. Okay? Remember that. But you have to study. You should know. And there is easy trick. See. All of you are aware of the classification of enzyme? Yes or no? Shall I recite it? Recall it? एक short revision किया जाए. Classification 1. Oxido reductase. What was the slogan I have given you? What was the slogan I have given you? Oxido reductase. Redox reactions. Someone is going to, if someone is going to donate something, someone has to be there. So you know it, no? Say na. Okay, fine. And the donor is going to be rich in terms of hydrogen. The acceptor has to be poor in terms of hydrogen. So if anything, if any component is releasing H+, who is supposed to accept it in majority class? A coenzyme. Who can be a coenzyme? Who can be in coenzyme for dehydrogenase reactions? NAD plus and FAD plus. याद रहेगा? NAD plus and FAD plus. Fine. Next, transferase. What is the role of transferase? Transferases are the enzymes responsible for transferring groups other than hydrogen. Yes or no? What are those groups? They can be phosphate. They can be phosphates. They can be other components also. Fine. Yes or no? A classical example of transferase is kinases. Yes or no? I have said one more thing. If you see an enzyme named as kinases, close your eyes and write. Close your eyes and write ATP or GTP as a donor of phosphate group, or vice versa. If you see any reaction where ATP, GTP are the donor of phosphate group, donating the phosphate group, then the enzyme name should be, close your eyes and write kinase. Yes or no? Yes or no? Fine. Next class, hydrolases. They break or split the bond provided by addition of water. Okay? Lyses. They split the bond but without addition of water. Yes or no? Fine. Next, isomerases. Isomerization. Okay. Next, ligases. Ligases are responsible for ligating two components. If in the presence of energy, ATP, the enzyme class is synthetase. There are many other enzymes. Carboxylase. They require enzyme. ATP. If no ATP is required, enzyme class is, subclass is synthase. I have told you, there is a difference between synthetase. I have written somewhere over here. Synthase and synthetase. Remember? All of you? Glucose is phosphorylated. Glucose is phosphorylated at the 6th carbon provided by an enzyme where the phosphate group is donated by ATP to convert into a component called as glucose 6-phosphate. Yes or no? Enzyme name should be? Close your eyes and write chi suffix. Yes or no? What should be the prefix? Look at the substrate. Add the name of substrate over there in the enzyme. Either it can be glucokinase or it can be hexokinase. Yes or no? Simple. Okay? If we talk about hexokinases, Okay. There are isoforms that stands from 1, 2, 3, 4. 4 isoforms are possible. Okay. Out of which 3 isoforms are readily responsible for splitting of glucose and other hexoses also, but specificity is for glucose. But other hexoses can be also splitted, or they can add phosphate group to other hexoses except glucose. Other hexoses also. Enzyme should be hexokinase 1, 2, 3. Okay? But hexokinase isoform 4 is responsible only for adding phosphate group to glucose. Only glucose. Yes or no? Okay? Okay? Importantly, isoform 1, 2, 3 are present in almost all the tissues. Almost all the tissues. But isoform 4 is present in liver parenchymal cells and beta cells of pancreas. Beta cells of pancreas. Okay? They are present in beta cells of pancreas. That means they are also going to play a role as sensor of glucose in the blood. What they are going to perform then? What they are going to perform? They are going to act as a sensor of blood glucose level. वो हाथी और चींटी वाला story याद है? Is that comfortable बेटा? वो that elephant or ant वाला चींटी कहानी story याद है? I had food like elephant. My insulin got released is like ant. What will happen? My blood will be flooded with glucose. Will remain flooded with glucose. It will start. The glucose will start coming out in the urine. No benefit. But if I had food like an ant, but my insulin got released as elephant. Tata, bye bye. Gone. The case is gone. Swarg Siddhar gaye. Who controls that? Remember the transporter? What was that transporter? GLUT2. Remember? It is present in beta cells of pancreas. I have told you that it also acts as a sensor of blood glucose level. It senses. Fine? Same like that, it is hexokinase isoform number 4 that is also called as glucokinase, that is present in liver cells and beta pancreatic cells. And that also acts as a sensor for blood glucose level. It is also responsible to sense the blood glucose level and release insulin accordingly. Yes or no? Okay? If, for example, now I have already told you about the affinity and km of GLUT2, if you remember. Okay. You are going to say me that glucokinase or hexokinase isoform number 4 will be having a higher affinity for glucose or lower affinity for glucose. Anyone? What I am saying, listen to me carefully. It is present in beta cell of pancreas. Beta cell of pancreas is responsible to release insulin dependent on the amount of glucose there present in the blood. Yes or no? Okay? In that scenario, the affinity of this enzyme for its substrate bole to glucose, has to be low or high? That तुम्हारे हिसाब से चलते हैं. चलो, I'll go with you people. You said high. हाँ बेटा, देखो. Oh, madam, देखो. You said high? No, you didn't say. Who said high? You said. Fine. Let us go with that. Imagine the affinity is high of this enzyme for glucose. Huh? You, all of you? No. Many of you didn't had breakfast. Many of you, even I did had breakfast. If my affinity of glucokinase would have been high, fine. At what condition right now we people are? Hypoglycemic state. Yes or no? If the affinity would have been high, affinity matlab love of the enzyme for the substrate. If the love is high, it will catalyze the glucose. It will convert the glucose into glucose 6-phosphate in the beta cell of pancreas. That will cause the release of insulin. You are already hypoglycemic and insulin is released. We will become supra hypoglycemic. टाटा बाय बाय। क्लास लेते लेते मैं यहाँ पे ढाक से गिर जाऊंगा, गॉन। Got my concept? So affinity cannot be high. अगर हाई होता तो हम लोग रोज रात को खाना खाते, 3 बजे तक हमारा ग्लूकोज मेंटेन रहता, 4 बजे के बाद टाटा बाय बाय, खत्म वहीं पे। रोज रात को वही होता। लेकिन ऐसा होता है क्या? रात को 10 बजे खाना खाते हो एंड एवरी मॉर्निंग यू राइज अप, yes or no? So the affinity of glucokinase provided for the substrate has to be low. That means this enzyme is going to act only when the concentration of glucose in the blood is high. याद है? Dairy milk and Kit Kat. You love Dairy milk? One piece is enough. You don't like Kit Kat. 100 pieces are not enough. No sir. मजा नहीं आया। क्या कंफ्यूज बन रहे हो? Yes or no? Is that clear? So, the affinity provided for glucokinase is going to be low. Affinity of glucokinase for the substrate, glucose, has to be low. If low, then what should be the Km? What should be the Km? Inversely proportional. Remember? Simple. Now, for other parts or other isoforms of hexokinase 1, 2, 3, the affinity should be high, because these isoenzymes are present majorly in tissues like brain cells. This enzyme is present in tissues like brain cells also, in cardiac muscles, many other tissues. RBC, which are responsible to which are dependent on glucose for the production of energy. Which are dependent on glucose for the production of energy. Yes or no? Okay. So if the affinity is going to be high, in that scenario, even smaller concentration of glucose is present in the blood, it will utilize. So to survive. So we are surviving. Is that clear? Sure. This, one more thing, whenever you see kinases reactions, close your eyes again and write Mg2 plus. Whenever or wherever you see kinases reaction, close your eyes again and write Mg2 plus. It has to be there as a cofactor. Why you know? This is all, ATP has got 3 phosphates. Huh? 1, 2, 3. That's why we call it as triphosphate. And the terminal phosphate is removed out. And the releasing of terminal phosphate is the responsibility of the enzyme to add a nucleophilic attack over the terminal phosphate. Now Mg2 plus is a positive ion that makes a shield of positive charge over the adenosine triphosphate, that allows the nucleophilic attack to be possible at the terminal end, causing the release of terminal phosphate. That's why whenever you are going to see kinases, you should write Mg2 plus as a cofactor. Okay. Reaction number 1. Reaction number 2. Reaction number 2 is converted into enzyme name, class, isomerases. Enzyme name, chalo, suffix is isomerases. Okay. Prefix, can we write it as hexose? No. There is something that is added to the hexoses, a phosphate group. Can we name it as phosphohexose? Think about the substrate. Suffix is already added. You understood it to be as isomerases. Now think about the substrate. Can we rename the substrate as phosphohexoses? Yes or no? Yes. So the enzyme name stands as phosphohexoses or hexose isomerase. Phosphohexose isomerase. Okay. Next reaction. Reaction number 3. Fructose 6-phosphate is converted into fructose 1,6 Bisphosphate. Why we add bis? Why we add bis? Why do we add bis? Anyone? अरे यार तुम लोग बहुत कम इंटरैक्टिव हो यार, मुझे अच्छा नहीं लगता. 2 phosphates are there, sir. Yes, sir. 2 phosphates are there. Okay. At position number 1 and at position number 6. We can add di, no? We can add di. Fructose 1,6 diphosphate because you have 2 phosphates. Why bis? I thought so. We add di where a phosphate group, when any component is, when any compound or any group is added upon a same carbon atom. Okay, but if the compound is added on 2 different carbon atoms, they are barely separated by each other, from each other, we add bis. Okay, can you see the phosphate group is present on the first carbon and the another phosphate group is added at the 6th carbon. So they are at 2 different carbon atoms. That's why we add the term bis. Okay, is that clear? Can anyone say me that is this process of different pathways are just occurring normally, or is there any significance? Why glucose has to convert into glucose 6-phosphate? Why there is, okay fine, you can answer it in the way like, sir, there is no direct enzyme which can convert glucose into fructose 1,6-bisphosphate. Okay, fine, good answer, fine. Compartmentalization of enzymes. Is there any other need of converting the glucose to glucose 6-phosphate? Anyone? You know, glucose being a polar molecule, glucose 6-phosphate is more polar. Okay? If we talk about the transporters present over here in the cellular membranes, they are not responsible. There is no specific transporter for the import or export of phosphorylated compounds. Remember that. They cannot import or export phosphorylated compounds. But the glucose that has come inside from by GLUT2 can readily move outside by using the same transporter. It just depends on the concentration gradient. Cell के बाहर concentration gradient was high, cell के अंदर the concentration gradient was low. As a result, glucose moved in. After some time, when the cell is rich in glucose, it can also move out. So the net profit is 0. Fine. So now, what the cell does? Cell adds a phosphate group over the head of glucose, which makes the glucose mobile. Can glucose go out now? No, because you have added a phosphate group. And as I told you, that phosphorylated components cannot move out of the cell. Rather, they can come into the cell. They cannot come into the cell also. Okay, so this step has got an importance. It is just like making the glucose stay inside the cell. This phosphorylation is required to make the glucose stay inside the cell. If you have come inside the cell, you cannot leave the cell. You are made to go and go under these reactions and convert into pyruvate. Fix. Okay, is that clear? Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. What is the difference between substrate and product? Arey, what is the difference between substrate and product? One more phosphate group is added. First carbon. Phosphate group added. Who should be the donor of phosphate group? AT. Fine. सही हो? क्या confusion है इसमें? 1 ATP is going to convert into ADP. Close your eyes and write. अच्छा, पहले काइनेस लिखे? अच्छा चलो ठीक. Closed my eyes and wrote kinases. Yes or no? Suffix. Again close your eyes and write Mg2 plus as a cofactor. Okay. What should be the prefix? Prefix. Fructose is phosphorylated. So phosphofructo. Fine. Phosphofructokinase. And this enzyme has got 2 isoforms. This is what's called as PFK1. Another isoform that is also present in cytoplasm is called as PFK2, that has got a very important role in regulation of glycolysis that we are going to talk later. Next class. Yes or no? ठीक है? If we talk about this red marked reactions, please remember it. We are going to discuss them in detail in regulation. They are going to help us in regulation. If we talk about the third reaction provided by PFK1, it is an allosteric reaction. Yes or no? Yes or no? Can we call it as an allosteric reaction? Allosteric enzyme? Are all enzymes provided in a metabolic pathway are allosteric enzymes? Are all enzymes allosterically regulated? Are yes or no? Karan sir has taken this class, no? What my question is? Are all enzymes allosteric enzymes? Then how to identify that this enzyme of a specific metabolic pathway is going to be an allosteric enzyme? How to identify? Sir, it is written in book, so I will mug off. No, not needed. You just have to look for the rate limiting step of an enzyme, of a pathway. Every pathway has got a rate limiting step. That rate limiting step of the pathway is going to have an enzyme that will be allosteric enzyme, and that has to be allosterically regulated. So for glycolysis, PFK1 is the allosteric enzyme because the third step of glycolysis is what we call it as rate limiting step of the pathway that decides whether the fructose 6-phosphate is going to go for glycolysis or the fructose 6-phosphate is going to go for another pathways or other pathways. Got my point? Just like glucose, it is trapped inside the cell. This can go for HMP shunt pathway. Many other pathways are possible. It can go for HMP shunt pathway. Fructose 6-phosphate can go for fructose metabolism. But once fructose 1,6-bisphosphate is formed, it is an irreversible reaction. It cannot convert back. This makes confirmation, or this gives a confirmation that the glucose that is utilized for the formation of fructose 1,6-bisphosphate has to convert into pyruvate. Confirmed. Thappa. Is that clear? So rate limiting enzyme. And if you see this terminology, books stating that this is the rate limiting step of this pathway, close your eyes and think about that this enzyme is going to be allosterically regulated, or this enzyme is going to be an allosteric enzyme. Is that clear? Okay. Next comes splitting phase. This is a splitting phase. Okay, fine. This is splitting phase. The enzyme should be a classification of enzyme में example दिया था मैं इसका. Splitting phase, it is breaking the molecule into two parts, and I have told you God is never going to be partial with our cell. We people are partial, but God is never partial. It has to be splitted into two molecules of 3 carbon. 6 carbon is converted into 3 carbon, equal, right? Fine. Splitting, breakdown, cleavage, responsibility of two classes of enzyme, either hydrolase or lysis. Did I add any incorporation of water into it? No. So it cannot be hydrolases. It has to be lysis. Example is aldolase A. Aldolase A. There is another isoform called as aldolase B that is provided in fructose metabolism, and deficiency of aldolase B is responsible for fructose intolerance. We will study that in fructose metabolism. Remember that. Okay? It is aldolase A that acts on fructose 1,6-bisphosphate. It is aldolase B that acts on a fructose component. Okay? And the deficiency, hereditary deficiency of Aldolase B is responsible for a disease called as fructose intolerance. Yes or no? Fine. I have told you that aldolase A is a class of enzyme, comes under lyases that use other mechanisms provided for breakdown of a component, that called as reverse aldol condensation. Reverse aldol condensation is a process name. Clear? The product should be glyceraldehyde 3-phosphate, and some of you call it as DHAP. Don't call it as DHAP. I have heard many of students calling it as DHAP. No, it is not DHAP. It is DHAP. Full form is dihydroxyacetone phosphate. In school terminologies they use this. I know. It is okay. But now you cannot say it as DHAP. It is DHAP. Dihydroxyacetone phosphate. Okay. Next reaction comes, the DHAP is reversibly converted into one more molecule of glyceraldehyde 3-phosphate provided by an enzyme called as C. A ketone is converted into an aldose. A ketosis is converted into an aldoses. 3 carbon, 3 carbon. Enzyme should be isomerases. Isomerization. You are just shifting carbon, second carbon to first carbon. That's it. Functional carbon as second carbon in ketosis, first carbon in aldoses. Simple. You are shifting. So isomerases. This is called as triose phosphoisomerases. So ultimately you are going to get 2 molecules of glyceraldehyde 3-phosphate. Okay? Yes or no? Fine. Okay, fine. At the last. Now, next reaction. This glyceraldehyde 3-phosphate is going to get, now listen to my words very carefully because I am going to come up with some questions for you. Glyceraldehyde 3-phosphate is going to undergo the first oxidation reduction reaction of the glycolysis. The first maybe. Fine. Glyceraldehyde, aldehyde is going to convert into 1,3-bisphosphoglycerate. You know the scheme, no? Alcohol oxidized to aldehyde, aldehyde oxidized to carboxylic acid. You know that? You know that? So by name only we can understand, sir, aldehyde converted into glycerate, glyceric acid, carboxylic acid. The condition should be oxidation. What happens in oxidation? Either addition of oxygen या फिर removal of H plus ion. So removal of H plus ion has to happen. What was the slogan? If something is going to get removed, someone is ready to donate, someone has to be there to accept it. Yes or no? Who should be the acceptor? A poor one. Poor one is called as NAD plus. Yes or no? Huh? It accepts the H plus and converts into. Arey हाँ या ना बोलो यार. Huh? What I have told you, if you see this sort of reactions, if you see the involvement of NAD, if you see the involvement of FAD, the enzyme name should be dehydrogenase. Why? Because oxidation का मतलब क्या हुआ? Removal of H plus. Removal of H plus in terminology, what we call dehydrogenation. So the enzyme name should be dehydrogenase. Simple. The suffix should be dehydrogenase. Yes or no? Add the prefix. Substrate name is glyceraldehyde 3-phosphate. Write it. Glyceraldehyde 3-phosphate dehydrogenase. Yes or no? कितना सिंपल है, देखो रटने का जरूरत है नहीं है भाई, बिल्कुल नहीं है. If you go through this way, not needed. Mugging off is not needed. Understand? ठीक है? But is my reaction complete? All of you have gone through glycolysis in your primitive days. Not primitive, in your previous years. Is my reaction complete? Where? What is lacking? What am I lacking? See the difference. 1 phosphate was at third carbon. Here you have 2 phosphates. one at 1 carbon, another at 3 carbon. So 3 में तो पहले से ही था. You have added one more phosphate group. Yes or no? But this enzyme name is dehydrogenase. Is dehydrogenase responsible for addition of phosphate? No way. Did I miss any enzyme name? It has to be kinases? No, because I have not written any sort of ATP as a donor or GTP as a donor. Did I wrote? That means some part, some functional part of this enzyme is responsible to add phosphate group, but the phosphate group is not coming from donor as ATP. Yes or no? The phosphate group is not coming from the donor as ATP. Then who can donate phosphate? It can be an inorganic phosphate that we call it as PI. Yes or no? Yes or no? Okay? Importantly, this inorganic phosphate, coming to the clinical significance. Time? Coming to the clinical significance. Very commonly seen, arsenic poisoning. Arsenic poisoning. Generally, arsenic poisoning occurs because of the pentavalent form of arsenic called as arsenate. Pentavalent form, arsenate. That arsenate is responsible to remove this inorganic phosphate out of the environment. It removes, chelates out. It chelates out. As a result, there will not be any formation of 1,3-bisphosphate, rather it only forms 3-phosphoglycerate. Yes or no? Fine. If you see, the next reaction is conversion of 3-bisphosphoglycerate into 3-phosphoglycerate by the production of an ADP. We'll come to that. This is the next step that we are going to see. But if arsenic poisoning will be there, this inorganic phosphate will be removed. As a result, the product formed will be directly as 3-phosphoglycerate. And in arsenic poisoning, due to arsenic poisoning, it causes the deficiency of ATP, deficiency of energy provided in the cell. If this arsenic poisoning takes place in more you can say vulnerable for arsenic poisoning is RBCs, because RBC totally depends on glycolysis for the production of energy. Fine. And if arsenic poisoning is there, this PI will get removed. It will get removed, replaced by arsenate. As a result, it will directly produce 3-PG. As it will directly produce 3-phosphoglycerate, they will lose this step. This step, the next step to this reaction is a very important step of glycolysis, because it is a first reaction where you are going to see the production of an ATP. Before that, you have seen utilization of ATP. Okay, yes or no? So this is the first step where you are going to see production of ATP. Okay? And if ATP is produced at a substrate level, we call it as substrate level phosphorylation. Otherwise, the production of ATP is responsibility of electron transport chain provided by, that happens in mitochondria. That electron transport chain allows OXPHOS to occur, oxidation and phosphorylation, oxidative phosphorylation provided by an enzyme, a complex called as ATP synthase, complex 5, that produces ATP. Okay, but if anyway ATP is getting produced at the substrate level, at the cytoplasmic level, at any other organelle level, we call it as substrate level phosphorylation. Is that clear? So if I say that the RBCs are now going to produce less energy, they are going to feel less energized in case of arsenic poisoning, you understood why did I say? Because you are missing this substrate level phosphorylation if arsenate replace the inorganic phosphate. Because as inorganic phosphate is replaced, you are missing the formation of this component, 1,3-bisphosphoglycerate. Okay? It directly forms 3-phosphoglycerate, leading to the missing of an energy production in the form of ATP. As a result, we can say that RBCs lack ATP or energy. Okay, please take the attendance. We'll continue this in the next class.